Aging is often thought of as a gradual decline, a slow erosion of physical resilience and cellular function. But the more scientists understand the inner workings of aging, the more they're uncovering hidden mechanisms that don't merely fade with time. They become actively destructive. One recent study, published by researchers at the Buck Institute for Research on Aging, highlights one such mechanism: the role of the enzyme ADAM19 in amplifying gut inflammation and cellular decay. While the research has been presented with some optimism, suggesting a path toward new treatments, it also uncovers a deeply unsettling reality. As we age, certain enzymes like ADAM19 don't just malfunction, they seem to turn against the body, unleashing biological processes that degrade tissue, disrupt immunity, and accelerate systemic breakdown. ADAM19, classified as a metalloprotease, may sound like a minor player in the complex symphony of aging. But in this study, it emerges as a central figure in orchestrating a harmful cellular phenomenon known as SASP, the senescence-associated secretory phenotype. Senescent cells, which stop dividing due to DNA damage and other stressors, are not inert. They become biological saboteurs, releasing a toxic cocktail of pro-inflammatory chemicals, enzymes, and signaling molecules that damage nearby cells and tissues. This secretory profile plays a major role in chronic diseases ranging from cancer to Alzheimer's to inflammatory bowel disorders. What the Buck Institute team found is that ADAM19 is a critical driver of this toxic secretome. It helps process and release SASP proteins, effectively turning aging cells into chemical factories of inflammation. That means the enzyme doesn't just participate in the process, it enables and amplifies it. In fruit flies, mice, and human cells, inhibiting ADAM19 reduced markers of aging and gut inflammation. But this finding, while promising in a technical sense, is also a red flag. It suggests that the aging gut is being actively corroded from within, not by pathogens or external toxins, but by our own biology, by cells we can no longer count on to protect us. The results in animal models are stark. Mice subjected to chemotherapy, a common trigger of accelerated aging and gut damage, had significantly better intestinal integrity when treated with a drug that inhibited ADAM19. That sounds like a win, but dig deeper and the implication is more sinister. Our medical treatments, combined with natural aging, may be triggering a chemical storm in the gut, one that opens the door to leaky gut syndrome, systemic inflammation, autoimmune flare-ups, and long-term metabolic collapse. These effects aren't cosmetic or minor. They're central to the cascade of decline that marks the later years of life. Worse still is the apparent stealth with which this process unfolds. The cells involved are not being destroyed. They are still present, still alive, still capable of interacting with their environment. But they're changed, reprogrammed into agents of deterioration. This subverts the traditional narrative that aging is a matter of wear and tear. It paints a darker picture, one where the aging process actively recruits parts of our own body into a silent rebellion against homeostasis. We're not just falling apart, we're being slowly dismantled from the inside. ADAM19 is a warning sign, not just a therapeutic target. The fact that its inhibition produces measurable benefits in multiple species implies that this enzyme is part of a larger, conserved biological pattern. And that means the threat it poses is universal. Humans, like flies and mice, are susceptible to this enzyme's role in exacerbating cellular damage. And unless it is addressed directly, the cascade of dysfunction it triggers will continue unchecked in millions of aging individuals. This also complicates the current push towards senolytic therapies, drugs designed to kill senescent cells. While those strategies are aggressive and fraught with risks, the xenomorphic approach proposed here, which merely dampens the cell's harmful secretions, is not without its own limitations. It may control the worst symptoms, but it doesn't eliminate the root problem: a growing population of dysfunctional cells that, if left to accumulate, could outpace the body's ability to keep inflammation in check. And unlike infections or tumors, these cells don't alert the immune system in a clear, targeted way. They lurk quietly, modifying the environment, degrading barriers, and reshaping tissue landscapes in subtle but devastating ways. Another troubling aspect of this discovery is its relevance to medical treatments widely used today. Chemotherapy, radiation, and even some antibiotics can accelerate the formation of senescent cells. The fact that Adam-19's destructive influence is magnified in post-therapy environments suggests that many life-saving interventions may come with a hidden cost, a slow but irreversible weakening of the gut barrier, and a rise in systemic inflammation. That has enormous implications for cancer survivors, older patients undergoing chronic treatment, and those with already compromised immune systems. Moreover, the gut is not an isolated system. Its breakdown leads to widespread consequences: the leakage of bacterial toxins into the bloodstream, a process known as endotoxemia, chronic immune activation, neurological decline, and even cardiovascular stress. Once the gut wall begins to fail, the entire body becomes vulnerable to inflammation-related disorders. If Adam-19 is a linchpin in that failure, then it must be treated not as an optional target, but as a critical node in the biology of decline. While research into Adam-19 inhibitors like betomastat shows promise, clinical translation is still far off. These drugs may carry side effects, and their long-term safety in humans is unknown. Additionally, modulating a powerful enzyme in the human body always carries the risk of unintended consequences. What else does Adam-19 regulate? Could suppressing it in one tissue lead to dysfunction in another? These are the hard questions that must be asked before celebrating too early. What this study ultimately reveals is not just an opportunity, but a danger. Our bodies are aging in ways we are only beginning to understand, and some of these changes are alarmingly aggressive. The discovery of Adam-19's role in senescence and gut damage is not just a breakthrough, it's a red light, flashing urgently over the biological mechanisms we've long ignored. If this enzyme is as central as it appears, then our current strategies for managing age-related illness are far too passive. We must not wait for symptoms to appear before we act. Aging is not a passive state. It's a series of active, damaging processes, and Adam-19 may be one of the main players on that stage.